I usually get the delight and honor of introducing our researcher. I'm usually totally unable to pronounce any of the work they do, so I just usually run in with like very sexy brain. Please welcome Dr. Blue. But this time I get to actually pass this off, this particular task, because there is an incredible connection in this room today with our guest speaker, Dr. Jonathan Irish. As you might see here is one of our amazing Terry Fox friends, Miss Alison Stoneman of Orillia, Ontario. Ali, as her beloved friends love to call her, joined us in 2001 as the run organizer in Orillia. She'd been involved for many years before that as a Terry's team member, as a team captain, as a fundraiser. It's truly an inspiration to us all. But her connection today is a beautifully personal and incredibly emotional way to welcome our next guest. And so, Al, do you have the microphone?
Yeah.
Oh, here. I'll give you.
She's got it. It's on.
Everybody hear me?
Yeah.
Al's got it. Alison Stoneman, take it away, please.
Thank you, Martha. So you know by my red shirt that I'm a Terry's team member, and Martha just told you that as well. So I have had a diagnosis of cancer. My cancer journey began on Halloween Day in 1996. Now I'm looking around the room and I know many of you weren't even alive then. But that's okay. I went to see my family doctor that day about a skin problem I had on my hand. And while I was there, I said, could you also take a look at this sore on my tongue? And I knew by her reaction that this might be something serious. So from there, she sent me to see an ear, nose, and throat specialist, or ENT, who did a biopsy. And he told me to go home and get my affairs in order. I was 31 years old. A couple of days later, he called with the news that I had tongue cancer. Thankfully, that same ENT referred me to my oncologist, who worked out of what is now called UHN, University Health Network. So this oncologist gave me back some of the hope that was stolen from me by the ENT. And of course, I leaned heavily on my hero, Terry Fox, to provide me with hope and courage too. So my oncologist let me know that there was a possibility of joining a clinical trial. And thanks to Terry Fox, I already believed in cancer research, so I joined the clinical trial. Which meant, in addition to the removal of the tumor and the surrounding tissue on my tongue, which in the end meant they removed 35% of it, I would have further surgery to remove all the lymph nodes from the left side of my neck and head, which is called a neck dissection. The thought was that the removal of the lymph nodes would prevent the cancer from spreading from the oral cavity to anywhere else in your body. And although there were a few rough days after surgery and during recovery, I was very lucky to have been chosen for the arm of the trial that involved removing the lymph nodes. I was also very lucky to only require surgery. I didn't have to go through radiation or chemotherapy. I was very grateful to have survived, and I was getting back to my life when I heard the news that a man named Michael, who had been diagnosed with tongue cancer at the same time I was, and who was the same age I was, had died. He died. That was a huge reality check for me. When you're going through your days of preparing for treatment and surgery, and you don't have time to think about your own mortality. You just do what you have to do to get through that day and the next and the next. So that was when it really hit me that this was a potentially fatal disease. So Michael didn't have the advantages I'd been given though. He wasn't treated at UHN. He wasn't treated by the team led by my oncologist. And he did not take part in a clinical trial or any type of cancer research. He did have surgery to remove the tumor and the surrounding tissue. So as a survivor, I give huge props to the clinical trial for my success and for my life. And I'm eternally grateful to have been referred to UHN and the oncologist who led the team who treated me. Every year on the anniversary of my cancer surgery in December, my husband Jim and I celebrate my cancer-free day. And on my 29th, which was last December, he had this hat made for me. Its designs are burned into it. Anyway, you can, Martha's gonna hang on to it if anyone wants to see it. And then every 5 years we have a party and invite others to celebrate with us. So Jim and I are starting to think about what we'll do this coming December for my 30th cancer-free celebration. So I'm so very thankful that my oncologist and the clinical trial have given me almost 30 years of life beyond my cancer diagnosis. In that time, I bought a house, I met and married Jim. I became an aunt to my amazing niece and 3 amazing nephews, bought a business, and I've had some amazing adventures. I've also been honored to lead a group of volunteers in hosting the annual Terry Fox Run in Orillia. To date, our event has raised over $1,111,000 for cancer research. Now let me get back to my oncologist. I saw him annually for 22 years, and then I saw him a few summers ago when my new dentist had a concern, which thankfully turned out to be nothing. My oncologist, in addition to leading and performing surgeries and seeing patients, has also taught hundreds or maybe thousands of students over his career. Any of the students that I've met that have trained under him over the years speak very highly of him and how much they learned from him. But that isn't all. This amazing human being has also been involved in making the way that cancer patients are treated, and he's been doing cancer research throughout his career. It's been very interesting and exciting to learn about the various projects he's been involved in over the years. So like Martha, I was a little bit nervous about the big words and titles and all those things, and I was afraid I'd miss something. So I went to Google and I did a Google search for his name, and AI would tell you that he is a highly distinguished Canadian surgical oncologist, reconstructive surgeon, healthcare executive specializing in head and neck oncology. But AI doesn't know him like I do. He's so much more. And it's my incredible pleasure today to share him with all of you. My oncologist is here, Dr. Jonathan Irish.
Thank you so much, Alison. And I also, just as a matter of fact, see about, 4 or 5 patients or family members that I've treated here. I feel like I'm almost running a clinic. I'm actually very famous with the most important people, which are my grandson and granddaughter, who are 3rd generation Terry Fox runners. So the merch table outside I'll be hitting for those extra small t-shirts, and that's really important. And I remember walking down, we have a farm in the Hockley Valley, and walking down the main street of Orangeville at the farmer's market on Saturday mornings and seeing the Terry Fox table. And it just shows you the grassroots of this organization. That's what makes it special. And so thank you. And I'm also quite surprised, quite frankly, that I've been invited back. I think about 10 years ago I was invited, and they had an icebreaker question before the speaker was me. And it was, what was your favorite chocolate bar? And I was going to come up and people were going, it's this, you know, it's Kit Kat because it's this, or O. Henry because it's this. And I was trying to think, and I was coming up and I said Crispy Crunch, and I couldn't remember the reason. But I dropped the mic, and I just instantly said, well, I guess my chocolate bar is Butterfingers. And then I realized, whoa, I'm a surgeon. And so I'm therefore also surprised that I still get patients. But again, thank you so much for the opportunity to speak, because what you do is incredibly important. And you know, in April 1980, Terry Fox started his Marathon of Hope. He ran 143 days, the equivalent of 26 miles every day for 143 days. Now, I was about to say I am a marathon runner, and I better say I used to be, as you can tell, a marathon runner. I've run probably around 10 marathons. I've run the Boston Marathon 3 times. Now, no, that's not the story. The story is I can't imagine running a marathon the next day, let alone 142 days after that marathon. Oh, and by the way, I have 2 legs. Terry Fox, the physical accomplishment of what he did, perseverance, dedication, tenacity, is incredible. But you know, that's not the story. The story is legacy. And that's why you're here today. That's why we are here today. And so, thank you. And what I'm going to talk about is our journey. It's a journey of research and discovery, but it's a story that could not be told without you, the Terry Fox Research Foundation, and the support that we have had over the course of many years. I hope I pick up the right button. Have you got my presentation? No. And so what I'm going to talk about, and yes, I do use some big words, and some of them I don't even understand. But
Butterfingers.
I'm not going to be invited back. So, but you know, when I first started as a surgeon in 1993, you know, the hands and the eyes and the knife of the surgeon were very simple. And they're very different today than they were 34 years ago. And I can tell you, they will be very different 5 years from now. I mean, who would have known that the hand guiding the knife can most often be a robot guiding a fine instrument through the back of the tongue, or removing a lung cancer, or removing an ovarian cancer. The progress that has been made has been profound. And if you actually think back to the TV shows of the 1980s and 1990s, where the doctor put up the X-ray on a backlit film panel and read the X-ray in 2 dimensions very clearly, we see cancer now in 3 dimensions, and in fact the 4th dimension being time, because it changes over time. That course of being able to see the eyes of the surgeon today being very different than they were 34 years ago. And we keep thinking about what's the next generation, what's the next gen of where we are going. And so 25 years ago, our lab started to think about how do we guide, navigate, and track surgical instruments in a very fine and complicated 3D space. I'm a head and neck surgeon. The progress that has been made has been profound. And if you actually think back to the TV shows of the 1980s and 1990s where the doctor put up the x-ray on a backlit film panel and read the x-ray in 2 dimensions very clearly, we see cancer now in 3 dimensions, and in fact the 4th dimension being time, because it changes over time. That course of being able to see the eyes of the surgeon today being very different than they were 34 years ago. And we keep thinking about what's the next generation, what's the next gen of where we are going. And so 25 years ago, our lab started to think about how do we guide, navigate, and track surgical instruments in a very fine and complicated 3D space. I'm a head and neck surgeon. Doesn't get more complicated than that. So our engineers started to create that 3D dimension, that dashboard for the surgeon shown here, where you're seeing a CT scan in the upper left and upper right, in a coronal axial, which is like this, a coronal, which is like this, and a sagittal, which is like this in real time. And you can see the crosshairs of the instrument. We're not using incisions, we use a small tube that passes through the nose to take out a tumor at the base of the skull. And you can see that our engineers created virtual reality, which is shown in the bottom right hand, the yellow showing the optic nerve. That's the nerve that supplies vision. The carotid artery in red, which is blood supply to both sides of the brain, and of course the purple being the tumor. All of these the engineers did by virtual reality, by removing the bone virtually so that we can see what's behind that bony wall. That concept of understanding where we are in 3D and in 4D space as we operate, that 4th dimension being time. That concept of navigation and tracking. And we were pretty good at what we were doing. Now down the hall, and over the last 15 years, and certainly supported by the Terry Fox Research Foundation, we knew that that next generation was coming down the pipe. And it really was the development of nanoparticles, nanomedicines. As soon as you put nano in front of everything, everyone's going, oh my God, what's going on? I didn't even pass grade 12 chemistry. Nano just means small, okay? And all nanomedicine and nanoparticles are man-made particles that we can make in the laboratory. Nano means they're small. We can see them under the microscope. They're about the width of a human hair. But they can be built, and we can build them specially. And that's what we've been doing. And a colleague down the hall from me was working on development of nanoparticles. And he created, and I'll be talking about the nanoparticle that we have created, and we have translated that into what we think will be the eyes, the hands and the knife of the future, of the future surgeon. And this is all this new emerging area of theragnostics. Another big word. What does that mean? It means molecules that have multifunctionality, ability to function as a diagnostic molecule and a therapeutic molecule. You put those two words together and you have a fancy word called theranostics. That's what we get paid the big bucks for, to make fancy words that no one understands. It's simply a diagnostic molecule that can also be therapeutic. That's cool. But it's more cool because we started to collaborate. Here I am, a surgeon, interested in navigation and tracking in 3D space and 4D space, and I'm collaborating with a nanoparticle engineer next door, and Terry Fox Research Foundation realizes that it's about teams. It's about teams coming together where the sum is greater than any one of its parts. And we started to think, hey, I have this, you have that, let's put them together. And so this concept of we're in an era of nanomedicine. I'm sure we've had surgery, and sure we've had radiation treatment and systemic therapy or chemotherapy, and now of course immunotherapy, but we're now in the early parts of an era of nanomedicine and nanoparticle and theranostic treatment. And now you can go home and talk about theranostics. So let's talk about the molecule that we created. It's called a porphisome. Don't get worried, it's okay. A porphisome is quite simply, and I have notes here to keep me simple. So it's a naturally occurring molecule. First of all, it's small. It's about 1/100th the size, the diameter of a cancer cell. Small, nano, remember? Nano, you knew that, it's small. It's also natural, meaning it's formed from a ring that is available organically, meaning it's formed from a porphisome ring. And what a porphisome ring is, you all have it because you have blood. A porphisome ring, if it has iron in the middle of that ring, means you have hemoglobin. And if you put a magnesium in the middle of that ring, it means your plant has chlorophyll. It's natural. So we took a natural compound that's available and we modified it. We added an attachment to it that was able to be activated by light. You don't need to know the chemistry around that. If there's questions about that, I'll phone a friend. The point is we can attach a molecule to the porphisome to make it therapeutic. The cool thing about the porphisome is it's fluorescent. You shine light on it, it's fluorescent. It shines green. It's diagnostic. We can see it. The eyes of the future surgeon. I can see where the cancer is because the molecule will go to the cancer. We shine a light on it, it fluoresces. That chemical attachment that we attach to this makes it photodynamically active, meaning it can destroy the surrounding cells. We have a theranostic molecule, a diagnostic and a therapeutic molecule all in one package. And we have to protect. Doesn't seem to be forwarding. We have to protect that molecule by packaging it in a liposome bilayer. What that means is a bunch of fat. We have lots of that, right? So, but that allows it to go to the cell, protected, be absorbed by the cell. We take out that capsule. We can then fluorescently detect it and activate it with the light. And by activating it with the light, we can destroy the cell that has taken it up. And that is what the porphosome theranostic molecule is all about. It's all about the eyes, we can see it. The knife, we can destroy the cell without a scalpel. And so this is the concept, the new concept of nanoparticle treatment and theranostic treatment. We can do other things. It's not only multifunctional, meaning both diagnostic and therapeutic, but we can do other stuff to that molecule. For example, we can add magnesium to make it seen on an MRI scan. We can add copper to allow it to be seen on PET scanning, which is another form of imaging. We can add iodine to it to allow it to be seen on CT scan imaging. As I said, it is photodynamically active, meaning we activate it with light to destroy the cancer cell that has taken it up, and similarly we can heat it up so that it destroys cancer. And we're working on a concept where we put drugs in the middle of that layer, in the middle of it, so that when the molecule goes to the cancer cell and we destroy it and unsheath the fat protective layer, it only goes to the cell. That cancer drug only goes to the cell that it is taken up, so that the rest of the body doesn't see the side effects of a chemotherapeutic agent. So these are the things that we are thinking of or doing right now. So the next step, of course, once you start to develop the molecule, is to test it. Is it safe? Does it work? And so this is a rabbit with head and neck cancer, a mouse cancer. You can see it on CT scan imaging. Sorry, this doesn't seem to be. You can shine light on it. You can see the tumor. You can see not only the tumor that has taken up the fluorescent molecule, but you can see the lymph node where the cancer has spread. And so this concept of image-guided surgery, and if you can let that, and you can see both, here's the tongue where the cancer was, image-guided surgery, and you can see the lymph nodes that have taken up the cancer. And remember, this is just the diagnostic part of the molecule. The opportunity of destroying, as I said already, remember these molecules are not just diagnostic, they're potentially therapeutic. So this is where we're gone, or where we have come from. So what's the next step? One of the biggest challenges that I have isn't just the breadth of the cancer, but the depth. The tip of the iceberg problem. And so there's a cancer of the tonsil, for example. That's at the back of your throat. Some of you might have had your tonsils removed as a kid, but that's a tonsil cancer. If I were to go to remove that, I can see the surface of the cancer. So I'm pretty good about judging where the margins of the cancer are on the breadth. But how deep is the cancer is a huge challenge. It's a challenge not just in head and neck cancer, but in every cancer surgery treatment. And so this concept of the tip of the iceberg and understanding the depth of the cancer is so critical. So I am very lucky that I work with 5 really smart engineers, and they started to think of, well, the reflection back of the fluorescent molecule is very dependent on the depth of the cancer. If it's a very surface-oriented cancer, the reflection is very different of the light coming back to the camera is very different than if the tumor is deep. And they started to create AI mathematical algorithms to basically tell us, using this work, about how deep the cancer is. So we get a feedback on a kind of a light reflection map of the depth of the cancer as we're operating, using the same fluorescent diagnostic molecule. So it's not just telling us the surface of the molecule, but the depth of the molecule. Well, that story would stop here, but remember, it's important to translate this ultimately to patient care. And that's a long and arduous process. Remember, we started this work probably in around 2011. And so what's the next step? Well, we have to get Health Canada to approve. There's a whole, and in a good way, there's a whole process of getting approval to go first in human. But I was thinking, well, wait a sec. There must be some models where we don't just go from here to human. And I was thinking that I knew dogs and cats, companion dogs and cats, get cancer, mouth cancer as well. And so we went to the University of Guelph, and they had problems. They have dogs and cats who get mouth cancer and would otherwise die because they can't survive surgical treatment. They're just too small to undergo the surgery. And so we started about 3 years ago. We started, actually 4 years ago, we started a clinical trial on dogs and cats, companion dogs and cats who get mouth cancer. And they get thyroid cancer and other cancers as well. And we started to create, first of all, does it work in these patients? And it also allowed us to create a human-like operating room where we have an anesthetist, where we have nurses, where we have a veterinarian surgeon. Michelle Oblak was a veterinarian surgeon in Guelph who collaborated with us. And we developed an approach of treating dog and cat patients who get mouth cancer. And the results were outstanding and allowed us to build more and more evidence to go in humans. And you'll wonder, why do we put goggles on these dogs? Well, you can appreciate that as the molecule circulates, it does go to other organs. It doesn't just go to the cancer, although preferentially does go to the cancer, but it goes to the eyes or retina. And so we have to protect the dogs and cats' eyes because of light, because light will activate and destroy their vision. Now is there any guesses about what we call these? Goggles, yes. Goggles. So, yes. And the results have been profound. And this is an example of a tongue cancer in a dog, and you can see very clearly where the cancer is. And by the way, notice that small section of cancer that is away from the large area of the green uptake. That cancer might have been missed, right? Diagnostic, eyes of the future surgeon. The concept here is that we can better appreciate and augment our vision because of this new technology, because of a man-made molecule that goes preferentially to a cancer. And so we introduce the laser fiber optic light into the tumor because these dogs and cats can't undergo surgery. They're just too small, and quite frankly, they can't tolerate what a human can tolerate, and it destroys the cancer without a scalpel, without an incision. Here's an example. You can see an MRI scan, again demonstrating the multimodality function of the molecule. You can see it on MRR. You can see it, the fluorescence uptake by the tumor, including the satellite lesion. And then, of course, you can see the introduction of the knife of the future surgeon, which is a laser introduced into the tumor to activate the molecule and destroy the cancer. that has taken up. This is a cat who had cancer on both sides of her tongue, and she would have died, unequivocally died, with surgery, but we were able to control her cancer for long periods of time. Now, you can't ask a cat how they're feeling after surgery.
They won't tell you
But
anyway.
what happens is with mouth cancer, they can't drink, they can't eat. And so we use outcome measures like, are they drinking? Are they eating? And are they wagging their tail? It's not very, very different from my human patients, by the way. But these are the kind of outcome measures you look at. So, the Marathon of Hope has been a long pathway for us. Yes, it has gone from when porphyromes were discovered, also understanding imaging and 3D imaging and navigation and tracking. And we put things together, we start to understand that there's a sum greater than its parts, that we can impact patients, including the concept of down that pathway of veterinarian studies to help our patient dogs and patient cats, using AI-powered imaging to tell us, and using machine learning to tell us where the margins and the depth of the cancer are. And finally, we've gone first in human. And we've now treated 3 humans, not with mouth cancer, but because we have to go safely and in a slow incremental process, we're actually using this in endometrial or gynecological cancer to just image the patient, not treat them, just image them, mainly to look at the qualities of safety, to make sure that the humans who receive the MET treatment are just as safe as when we have treated our dogs and cats and other animals down that pathway of discovery. So this pathway has been a long one. Nothing like the Marathon of Hope. And when I was in 1980, it was July 13th, Sunday, July 13th, I was actually canoeing, training for the national championships with my crew in 1980, looking up at the bridge as he was crossing, very moving. And I have to say, when I was invited to give a speech in Oakville at the volunteer workshop for the Oakville run, I gave it literally hundreds of meters away from where I saw Terry 40 years earlier. So really important work that you are doing. You know, the overall survival of cancer, when Terry ran his Marathon of Hope, was about 40%. It's now around 70%. Moving that needle has been slow, incremental, building blocks upon building block. It's not going to be a eureka moment of cancer is cured. It's going to be building block on building block that will enable this, and you are part of those blocks. We need to make sure that cancer is a word and not a sentence, and I really want to thank you. Best put by Terry, right here. Even if I don't finish, we need others to continue. It's got to keep going. And it's the legacy. That's the story here. The legacy that we want to thank you. Best put by Terry, right here. Even if I don't finish, we need others to continue. It's got to keep going. And it's the legacy. That's the story here. The legacy that we all carry. And so thank you on behalf of my team, the inventor, Gang Zheng, of the molecule, shown in the middle, Brian Wilson, who is probably the world's expert on fluorescence, myself, the Sesame Street, which one doesn't belong to certain? But we're all on the same page. Let's finish it. And thank you. I want to thank you. Best put by Terry right here. Even if I don't finish, we need others to continue. It's got to keep going. And it's the legacy. That's the story here. The legacy that we all carry. And so thank you on behalf of my team, the inventor, Gang Zheng of the molecule, shown in the middle, Brian Wilson, who is probably the world's expert on fluorescence, myself, the Sesame Street, which one doesn't belong to certain? But we're all on the same page. Let's finish it. And thank you. Wait till I show my grandchildren.
Dr. Irish, Alison could not have been a more beautiful introduction for you. All I can really say is thank you. You will always be the biggest brain in this room, so you're welcome back anytime. But we have, I think most people might know, hopefully, that the purple shirt is a very precious color for the Terry Fox Foundation. It is for our researchers. Because we felt a few years ago that it was just as important as for our red shirts to exemplify Terry's team, that our purple shirts would exemplify the people who are working for the best phrase I think I might have ever heard, to make cancer a word, not a sentence. So thank you, Dr. Irish, for everything. So amazing, and I think quite serendipitous. You have a dog bandana in your package, and now I have to walk back my dog comment. Thank you to those dogs and cats, for their amazing contribution. And I think my brain is always blown by the researchers, but to know something about the work that's done, the long journey, but that it takes us to places I don't think any of us could have imagined. So thank you guys for every dollar you have raised has made Dr. Irish. Just spectacular. And I do want to say, I don't know if, oh, there's Michael. Michael Mazza. I want to say that Michael has, we have had a long legacy of talking about Terry and our cancer research, but I want to say thank you to Michael for the amazing work you have done with Terry Fox Research Institute as our CEO, connecting these two organizations with Dr. Jim Woodgett, who has taken over. And I think we are knowing more about the work we do, the progress, the importance of the Marathon of Hope Cancer Center networks than we ever have before, and we get to see this kind of work come to life that impacts every one of our lives. So thank you for that too, Michael.